Now, you told me that you got confused with coordination. Some things, yeah, it is okay. You were right, because this cell biology actually it is the basis of biology. Okay? Someone was asking me that what should I read first. I told them cell biology. Why cell biology? Because it is the basis of biology. All these other things, this physiology of animal, because all these biology topics, they are the life processes of animals. All these other life processes, they depend on cell biology. Talk about respiration, talk about photosynthesis, talk about coordination, chemicals of life, protein synthesis, eh? Maybe ecology, interaction of animals, maybe that one. Talk about locomotion, talk about growth and development. So everything rotates around a cell. Okay? So you need to know what that cell is. What is a cell? You may feel it to be obvious, but that cell actually it is way way deep than even up to now people are still discovering new things concerning a cell. Okay? So you never know.
Yeah.
You can also do research, you can also get interested in a cell and do research and become a professor in cell biology. There is a lot that is not yet known as far as a cell is concerned. Okay? So welcome to cell biology. So a cell, as simple as you know, all living organisms are made up of cells. Like you are there, don't you have cells?
I do.
You do. Whatever you are able to do, actually, it rotates around the cells. For example, you are able to read that word, a cell, because they are cells that are responsible for perception, and then they carry out different physiological changes. And now that is coordination. Okay? To bring about the effect for you to see. So, in other words, we have different types of cells, and these different types of cells in our body, they perform different functions to bring about the effect. So before you think about any concept in biology, you need to know what a cell is. Okay? The basic structure of different cells, the organelles that are present in the cell, and actually each organelle, how each organelle, what is the function of each organelle, and how each organelle is adapted to perform its function. So we shall describe all that. By the end of this topic, cytology, cell biology, you will be a king, you'll be a professor as far as a cell is concerned. Okay? So, a cell, just like in senior one you studied that matter is anything that occupies space and has weight, and matter is made up of smaller particles, and you named those particles atoms. You remember that? Yes. So, now a cell is identical to an atom, but now for this one, it is present in living organisms. So that cell is the simplest collection of matter that can live. It is that simplest particle of an organism, okay? And different cells, very many cells add up to bring about an organism that you see. Now, the cell structure is related to the cell function. Depending on a particular function of a cell, it will determine its structure. For example, the nerve cells, the neurons, they are different from muscle cells and different from the rod cells in the eye, the cone cells in the eye, and different from sperm cells and the ovum, and different from macrophages, okay? And different from red blood cells, and so on, and so on, and so on. Even when we talk about plants, the different cells that make up the plant structure, the plant body, they are different in structure and function, by the way. For example, you can talk about the guard cells, totally different from epidermal cells, and totally different from cortex cells, totally different from mesophyll cells, and so on, and so on, and so on. Even when we talk about lower organisms, protists, they are they are unicellular. Yes, we are going unicellular. We are going to unicellular. But being unicellular, they also have different structures in their cells. And having different structures, meaning they can inhabit in different environments. They do perform different things. For example, if you consider amoeba, it is different from Euglena, different from Chlamydomonas, different from from protos, this Plasmodium, and so on, and so on, and so on. Even when you talk about bacteria cells, they are different from these other cells. How different? We are going to see. They are prokaryotes. And so being prokaryotes, it means they totally perform. Yes, some physiology might be the same, but the cells are different. Okay? So depending on the cell structure, it determines the cellular function, the function of a particular cell. Okay? Not all the cells are the same. Now, all the cells are related by their descent from earlier cells. We are going to see the cell theory. And actually, this is almost a cell theory, which says that all organisms are made up of tiny particles called living cells. And living cells come from pre-existing cells by cell division. In other words, right now you cannot create a cell, but rather cells are formed from other cells. And however, it is believed that these other cells, the ones we assume that they are advanced, they come from, or they came from the, the, the other primitive cells. Okay? And actually this concept, it is a good evidence of evolution. Okay? So all these cells, starting from bacterial cells up to your cells, they have things in common. Okay? For example, if you are to consider, they all have the DNA. As you go up, all prokaryotic cells have mitochondria, all of them. And did I say all prokaryotic cells? Sorry for that. All eukaryotic cells have mitochondria. They all have membrane-bound organelles. Okay? All plant cells have chloroplast. And if you are to consider their biochemistry, if you are to consider the chemistry that occurs within these cells, for instance, respiration, it is the same, right from the primitive ones, the so-called primitive cells, up to you advanced organism. All your cells, they do carry out the same respiration. They all do carry out the same replication. Okay? The same protein synthesis. Though there might be differences, but the differences are minimal. Minimal. For example, we see that in replication, prokaryotic cells form replication bubbles, but eukaryotic cells form a replication fork. But that one is just a mere difference due to the differences in their chromosomes. But the rest is the same. So why is it the same? Probably they all came from same cells. And because of their different environments, then they evolved to suit their environments. Is that okay? So the study of structure and function of cells in plants and animals is what we call cytology. So if you might find cell biology or you might find cytology, it is all the same thing. As long as you are studying a cell, there is a lot to do with a cell. Forget the other senior one work, where you just drew a cell, this is a plant cell, this is an animal cell, and you just gave few differences and stuff like that. Here, there is a lot to do, to study, as far as a cell is concerned. Is that okay? A question?
Yes.
As far as cell is concerned. As far as what we've talked about is concerned.
No, not yet.
Not yet. Why don't you bring that chicken?
It's okay.
It is okay. Now,
Yeah.
can you see a cell with your eyes?
No.
Obviously, no. So, as early as 17th century, nobody knew. Actually, little or nothing was known to the cell world. Okay? Because
Yeah.
they didn't have anything to use to see this so-called a cell. Not until I think he's called Robert Hooke. He was the first person to hypothesize something to do with a cell. Something. He was the first person to throw light on the cell world. Okay? That they are cells. So I don't know. I think before him, cells never used to exist. Like there is this saying that before which science? Which guy? Before Newton, yeah, before Newton, things used to just move up with there was
Not.
no gravity. So before Newton there was no gravity. So things just used to move up, eh? Like you throw a stone, it just moves up. You kick a ball, you watched which move is this? Shaolin soccer, eh? You kick a ball, it just
Doesn't
goes
come back.
and doesn't come back. However, the ball in Shaolin soccer came back.
After a long time.
So that was before Newton, eh? So also before Robert Hooke, there was nothing like a cell. So I don't know. Maybe organisms. I don't know. I can't even think about it. Okay. So it was this Robert Hooke who hypothesized that there is something called a cell. So you can imagine, actually, that is before if you are still in P7, you don't know anything there. Do you study cells in P7?
No, no.
Because I also remember I was introduced to the cell world in my senior one.
Yeah, it is
So you
true.
can, I think before Robert Hooke, all people had knowledge that is equivalent to our P7.
We watch documentaries, so no.
So you can imagine. But anyway, so after that guy hypothesizing, then there is his friend. He was called, is it Reno? Reno Hill? Reno Hill, something of that kind, eh? I've forgotten the other name. Now it is this friend who tried to invent a microscope, okay, that really confirmed that there is a cell world. And actually, this the Robert Hooke was, he gave a visit to this Reno Hinock something, and so when they jazzed, when they discussed, then they were able to come up to a conclusion, to come up with a conclusion that there is a cell world. Okay,
Yeah.
so why that? Because these cells are not, they are not seen with our eyes. So if you don't have something to magnify, something to take you to the cell world, to that virtual world, then you cannot, you cannot know that there is something called a cell. Not so?
Yeah.
So therefore, this microscopy, it is some big concept as far as cell biology and biochemistry are concerned. Okay? So these cells, they are usually too small to be seen by our unaided eye. Okay? But the development of instruments such as a microscope, okay, that extended the human senses allowed the discovery and studies of cells. So before this, we didn't know anything to do with a cell. So this is a microscope. This is not the first microscope, but it is one of the oldest microscopes. Okay? So it is this microscope which tries to magnify. It is made up of lenses that when you put here your cell that you cannot see with your eyes, then the lenses within this microscope, the objective lens and the eyepiece lens here, they will magnify this tiny organism so that you are able to see it with your eyes. Okay? And you get amazed with the cell biology. So to study cells, biologists such as Utim use microscopes and other tools of biochemistry. There are many tools, not just a microscope, but to study cells, to enter into cells and do enough research, enough study, then you must have such instruments. Okay? Then we use microscopes to visualize cells that are too small to be seen by our naked eyes. Now these microscopes, they are divided into two types. And the first one is the light microscope. Now this one, it uses visible light that passes through specimens, okay, through a given specimen, and then through glass lenses. And now these glass lenses, they do magnify the image so that you are able to see your specimen. Now you don't see the specimen, but you see its image. Okay? So light must first pass through the specimen and then through these glasses, these lenses that will magnify the specimen so that now your eye is going to view something. that is clear. Now the clarity, the quality of the image, it will depend on the following factors. Number one, the magnification of that glass, of that lens. Now magnification means how big, how the glasses are going to magnify, okay, the specimen for you to be able to see. I assure you, in that environment where you are, you have a lot of microorganisms, very many of them, in that environment, in that air that is saturated. But because they are too small, they cannot be seen with your eyes. The eyes have just one lens, and the lens just does the refraction of light rays to the is it retina? Yeah, not cornea, to the retina, so that you form an image, which is now the image is now converted into a nerve impulse, which is transported to the brain for you to know that I've seen something. But now these microorganisms, they are very small that you cannot see them with your eyes. So when light that has passed through these microorganisms passes through these lenses, the lenses are going to magnify, okay? And depending on how many lenses that light has passed through, it will determine how big an organism is going to be, okay? So the magnification, it is the ratio of the object's image to the real size of that object. For example, if this is your object, okay, and now this object, light from this object passes through maybe one lens, and then this is one lens, maybe this is objective lens, okay, and then the light is magnified, okay, and you are able to see the object, and then this light passes through another objective lens, sorry for that, another objective lens, and this light is further magnified so that you see a bigger image, okay. Maybe this is your image, my screen is over, this is your image, oh my god, so this is your image. So to get the magnification, you measure the size of the object, of the image, okay, size of the image size, and then you measure the size of the object size, okay, and then you get the ratio. If the object maybe was 8, and if the image is 8, maybe 8 centimeters, and the object maybe is 2, maybe 2 centimeters, then the magnification is going to be times 4. Now what does this times 4 mean? Now don't put centimeters, it doesn't have units because it is a ratio. Now this times 4 means the image is 4 times bigger than the object. Is that okay?
Yes.
Now how about if it is times a half? You can write this times a half, or you can write times 0.5. It is all the same thing. Now if it is this, then it means the image is half the real object. The real object. Is that okay? Now how about when you use a microscope? Now when you use a microscope, okay? Remember, microscope has two lenses, because you might be in a lab and you are tasked to use a microscope. It has two lenses, where it has the objective lens and then the eyepiece lens. Now the objective lenses, there are 3 of them. There is one, there is another one, and there is another one. Now they have different sizes, where it has the raw power, and the raw power is usually times, is it times 5? Yes. Then no, it is times 4. Then the objective lens, there is high power and middle power. Middle power is times 10. Then low power, the high power, is it times 40? You check. And then this, the eyepiece, it is times 10. So if you want to measure, when you put your specimen on the stage, on the slide, and the light passes through this, okay, so this is your slide and you have their specimens. So the light that is passing through, it will pass through the specimen, through the objective lens, then through the eyepiece lens, finally through your eye. Now if you want to measure the magnification, then it is this, the objective lens, the magnification of the objective lens times the magnification of the eyepiece lens. So in this case, if this is 40, you check this, I'm not certain with this. It will be 40, then times the eyepiece lens, which is 10. So the magnification is going to be times 400. Okay? Times 400. So it is as simple as that. Now this means that the image is 400 times the real specimen, the real object. Is 400 bigger than the real object? Is that okay?
Yes.
So that is magnification. That's what magnification is all about. So if I draw a theme on the paper, I can also ask, what is the magnification? You should be able to tell the magnification. Then the quality also depends on the resolution. Have you ever heard of this term?
Resolution
Yeah.
in cameras.
On camera.
Yes.
Okay, that is good. That is good. Actually, yeah, that's good. Now, resolution refers to, actually, it is the clarity. It is also measure of clarity. And it depends on the shortest distance a given lens or a given instrument or a given, okay, your eye also, the shortest distance between two close points that an instrument can see as two separate points. Okay?
Pardon?
For example, let me use this. Are you seeing this and this?
Yes.
So this instrument, let me say if it is a microscope, this microscope, it views this as one point. You agree with me?
Yeah.
But this one, it views this as 2 points and the two separate points. You agree with me?
Yeah.
Now, this one has a high resolving power than this. You agree with me?
Yes.
Now, that shortest point where a microscope, or that shortest point between two close points, okay, where a microscope can still distinguish those 2 points as two separate points, refers to its resolving power. Is that okay?
Yes.
And the shorter the distance, the higher the resolving power. You agree with me?
Yes.
Yes. For example, I can put this point here and I put this point here. Now, do you agree that those are 2 points?
Yeah.
But
There are 2 points.
are you seeing them as 2 points?
Where?
This one here.
It's 1 point.
You are seeing it as 1 point
Yes.
because that is your resolving power. But if you bring a magnifying lens, or if I try to magnify, let me even try to magnify. Now, are you seeing that?
Yes.
Are they still, is it still 1 point?
No, they are two.
They are two now. So that is your resolution. So that ability to distinguish two cross points, okay, as two cross points, sorry, as 2 points is what we call resolution. And the higher the resolving power, then the higher the clarity, the higher the quality of an image. Okay?
Yes.
The higher the resolving power, the higher the magnification. For example, if microscope A has a higher resolution than microscope B, then it means microscope A can see tiny organisms compared to this one. Okay? Yeah, is that concept? Has it entered?
Yeah, they has entered.
Yeah, that's pretty good. Then there is contrast, and now this contrast is easy. It is the visible differences in parts of the sample. For example, if you are viewing maybe a cell, okay, and in a cell, this is, let me try to sketch. You are viewing a cell. Now, in one microscope, you might just see just a tiny circle like this. Okay? Then in another microscope, you might be able to see that circle with clear demarcation, even with a circle with some dense part inside this cell, okay, with other tiny parts, okay, with other tiny parts. Is that so?
Yeah.
Now it means that these two microscopes, this one has a high contrast and this one has a very low contrast.
Contrast.
Okay?
Yeah.
Now the contrast is brought about by the differences in light rays that are passing through this structure. And so for you to increase the clarity of an image, for you to increase the quality of an image, then you have to enhance the contrast, okay, of that image. Is that okay?
Yes.
Now the question is, how do you enhance the contrast of an image? By staining. Is that okay? So in biochemistry or in cell biology, there are different techniques that we use to enhance contrast, okay, of certain cell components. And one of them is staining. And we are going to see, there is this picture. Are you seeing this? So now in this picture,
Yes.
we just gotten cells, put them on a slide, and we mounted them on a microscope. And this is what we were able to see. But there is nothing you can conclude from this. There is nothing. So to enhance the clarity of this image, then you do the staining. And so when you stain, okay, then this is the image that you see. Now, aren't you able to see clearly that this cell is like this? Then there is some dense particle, dense
Yeah.
tape in the middle of this cell. Aren't you seeing that?
I'm seeing.
Now that is the concept of staining. That when you stain, okay, then you're going to enhance. Now, how are you going to, now in this, the light rays are passing through this equally. And so you want to be able to tell the clarity of structures within this cell. However, now when you stain, certain structures of this cell are going to absorb that stain and others won't. And so the difference is in transmittance of light rays through this cell is going to be significant, in that the light rays, for example, here they will just pass through here. That's why this is clear. Not so?
Yeah.
Here, some light rays will be absorbed, but here almost no light rays will be absorbed. And so you'll be able to clearly see this part. And you'll be clearly see this part. And so from this now, if you increase the magnification, if you increase the resolution of the microscope, maybe use another microscope with a high resolving power, then you'll be able to magnify this. Like if I try to magnify, you'll be able to magnify this and see what is inside. Now aren't you able to see this one, these ones? Aren't you able to see
Yeah.
them? You are able.
I'm able.
But in this other one, you can't see anything. Is that okay?
Yeah.
So in cell biology, microscopy is key. And so we use different stains. We use different staining techniques, okay, to study different structures. For example, there are different stains that are known, that if you want to study a nucleus, then you use this stain. It will stain a nucleus, okay, and it won't stain other structures, so that you are now able to see a nucleus. If you want to see organelles within a cell, then you will use this stain, maybe methyl blue. You use that stain, okay, and so you are able to see the organelles within a cell. If and this concept of staining, by the way, we now use it to determine the different types of bacteria. We have the gram positive and gram negative. So it is this staining technique that we are able to detect whether this one is a gram positive or a gram negative bacterium, so that now we treat accordingly. If it is gram positive, then you treat it accordingly. If it is gram negative, you treat accordingly. Is that okay?
Yes.
Yes. Now, most subcellular structures, including organelles, okay? Organelles, these are structures that are found inside this cell. They are too small to be resolved by a light microscope, okay? For example, the light microscopes that we have here in the lab, I can't take you there to show you a nucleus. Okay, you might see some dense structure in the middle. Like you might see this, but now if you see this, what will you be able to conclude? And what is this? So, to resolve this, then you need a better microscope with an enhanced resolution, okay, to be able to have detailed structures. And by the way, because of improved technology, that's why I tell you that we are now discovering new things as far as acid is concerned. And so that takes us to another type of microscope, and it is an electron microscope. Is that okay?
Yes.
Now the difference with a light microscope, for the light microscope, for it uses a beam of light. Light rays, it bases on light for its functionality. And you might have different sources of light, where one might be sunlight and those are the microscopes that you must use. You look at the source of light, where light comes from, you point the reflecting mirror, so that the reflecting mirror focuses that ray of light to the condenser, and then to the stage, then through the specimen to the lenses, up to when it reaches your eye. Or we have other electric microscopes. They are light microscopes, but however here the source of light is power. They have some bulb down that you connect it in a socket, you turn on, okay, and then you focus that beam of light through a condenser, then through a stage, through specimen, objective lenses into your eye. But the concept is one: light, a beam of light. Now, but now here, this one is kind of different, okay? That instead it uses a beam of electrons. So it focuses a beam of electrons through a specimen or onto its surface. Now those are the two different types of electron microscopes that we are going to see. You may have an electron gun that is going to shoot a beam of electrons, okay? Now, this beam of electrons, it will pass through a specimen or onto the specimen, and then through different electric glasses, that will now keep magnifying the specimen up to when this beam of electrons focuses onto, or is focused onto, the screen, and then you are able to see a clear micrograph. A micrograph is an image, a clear image of the specimen, okay, onto the screen. Is that okay?
Yes.
Now, the resolution is inversely related to the wavelength. Now, the resolution of any microscope is inversely related to the wavelength, okay, of light or electrons, depending on the microscope that you are using. Now, the wavelength of electrons, okay, is way shorter, much shorter compared to the wavelength of light. So meaning these electrons travel at a high speed compared to a beam of light. And so if the wavelength is inversely proportional, meaning the shorter the wavelength, then the higher the resolving power. Okay? So as a result, if you want to view a clear image, then you have to use an electron microscope, which uses a beam of electrons, okay, with shorter wavelength compared to a beam of light. So the modern electron microscopes, the ones that we have right now in Uganda, as if we have one, and the one
One.
is, yeah, one. Is it a miracle? Is it a surprise?
Yeah.
Okay. They're
We
expensive.
have the Uganda Virus Institute,
Mm-hmm.
and I think viruses cannot be seen by a normal what?
A normal microscope.
Yeah, because they also just found out of recent.
Do they see? Do they see viruses?
Yeah, I think.
No, they don't see. They use PCR.
PCR.
Yeah, polymerase chain reaction. That test does not see viruses, but you detect their presence. You detect their nucleic acids. But you study that as if you continue with biology, you do medicine, or you do molecular biology. Actually, that is molecular biology. So interesting, so interesting, so interesting. Okay? Actually, all my interests are in molecular biology.
Molecular.
Yeah. So it's so interesting. But they don't see viruses, they detect. by PCR, polymerase chain reaction. So the modern electron microscopes can theoretically achieve a resolution of 0.02 nanometers. This is theory. Imagine 1 nanometer, okay, is equal to 10 power 9, actually 10 power negative 9 meters. Can you imagine this? So theoretically, it is assumed that you can see if you have your 2 points, okay, two separate points, and the two separate points have a distance of 0.002 nanometers, you can still see them as 2 points. Can you imagine?
Mm.
Yes, that one. But practically, this is what you can see. The other one is theory. Practically, you can see this. So the 2 points that are separated together by a distance of 2 nanometers. Do you see how small a nanometer is? So these microorganisms, usually their sizes range from 10 micrometers, okay, to 100 micrometers. But viruses, for them they are small. They are smaller than these viruses. But normal microorganisms, these ones which are small, they range from 10 to 100 micrometers. But this one is in nanometers. 1 nanometer is equal to 10 power 3 minus, because it is smaller, micrometers. Okay? So it means with this resolution, you can clearly see something with this size. Is that okay?
Yeah.
Yeah, with this resolution, you can see clearly something with this size. And so, due to this technology, this one, then we are able to study cells with ease. Okay? We are even able to enter inside a cell and see what is there. See all the organelles, all of them. Even go deep the nucleus, even deep the nucleus, and see what is there. Why? Because of this resolution.
Can it see DNA?
Yeah, why not? We know the structure of DNA. It is known. And we shall study DNA. Didn't I tell you in the beginning?
Yeah.
Hey,
Even those the bases, those small things inside, those
yes,
small things.
we know the structures of these organic bases. We know them. They are known. How the atoms are arranged. They can even be synthesized in a laboratory. So they are now known. Okay?
Yeah.
Hey. And why are they known? Because of this. Okay? This resolution. Is that okay? Any other question?
No.
So that is pretty good. So it is 100-fold improvement over the standard electron microscope. So this one is really enhanced compared to our light microscope, which just uses a beam of light. Okay? So electron microscopes basically have two types. The same, or the scanning electron microscope, which now this one, it just focuses a beam of light onto the surface. Okay? This is just the difference. This one just focuses a beam of light onto the surface of a specimen, and it just provides a 3D structure of the specimen. So here, if you are just interested in the morphology, in the outside, how the organism looks like, just how the specimen looks like outside, then you use the scanning electron microscope. Is that okay? For
Yeah.
example, there is this. This is the scanning electron microscope. This one. So you put your specimen inside. If you want, you can find out its operation. That is more than our syllabus. Now we don't focus on how it works. Okay? But if you want, if you are interested, you can read about it. So you insert your specimen inside, and then it is made up an electron gun somewhere. Then you put your specimen here. Then this electron gun shoots a beam of electron that is going to pass through the specimen, then through series of glasses that will keep magnifying this beam until when it lands onto a screen. So this beam of electron, it will pass through these glasses. Remember, it will first pass through the specimen and then through the glasses, okay, until when it lands onto the screen. Now this is the screen. Now this screen, unlike the light microscope where you view directly with your eye, here these electrons, you can't just put your eye there unless you want to lose your eye. You can't just put your eye there. So here it projects a micrograph on a screen. Okay, you'll be able to see that picture. You'll be able to see the specimen on a screen. Okay, and the image is going to be really enhanced. Is that okay?
Yes.
Yes. So that is basically. Now, this one, it is the scanning electromicrograph. Now, if you visit those books, the books I added to our class, you usually see
Okay.
the same. They will write same. Okay? Same. Then, same means scanning electro microscope. Now, if it is a picture, it will be scanning electro micrograph. Micrograph. Okay? Now, a micrograph, it is the image, okay, that is formed by a scanning electron microscope. So those books you usually see same of then they. So this is the cilia, the external surface of ciliated epithelial cells. They have cilia on their surface. But if you use the same, then you just see it focuses a beam of electrons onto the surface. Okay, so you just see the surface. But when you use a transmission electron microscope, a TEM, you can shorten it as TEM, then for it focuses a beam through, okay, the specimen. You see what is inside a specimen. Okay, so you can see that transmission electron micrograph, these are the same. This is a same, but this one is a TEM. So this one, you see what is inside. Okay, so you notice the difference.
Yes.
Yeah, but the working principle is all the same. They all use a beam of electrons.
Electrons.
Okay,
Yes.
yes. So now you have assignment here to do from that basic background that we have received as far as electron microscope and light microscope are concerned. Then I want you to compare, okay, the electron microscope and the light microscope. And with comparing, you give differences and similarities. Similarities means things they have in common. Then differences, things that are different. Now here, you are free to use a table. Okay?
Yeah.
Then you give the advantages of the electron microscope over the light microscope. Why would you prefer to use the electron microscope over the light microscope? But also the advantages of the light microscope over the electron microscope. Is that okay?
Yes.
Then finally, from what we have discussed, can you compare, did we? From what we have discussed, can you compare the electron? It is the same thing. It is the same thing. Okay?
Yeah.
Yeah. So literally, can I talk about this? What is the time? Okay, now let me talk about this so that we call it a day. Now you may be wondering that with cell biology we shall study the cell organelles. By the way, you need to make a list of cell organelles. Okay? They are very many.
Yes.
Now,
I only know the Glucai apparatus.
you know
The Glucai apparatus.
the Golgi apparatus, which is okay, but they are very many. Okay? And more are still being discovered. Now, how are we able to isolate these organelles to study each independently? We use this technique of cell fractionation. And what's up with this technique? For example, actually, I think I talked about it in whatever. Did I teach you transport? No. Now, we study different components of blood: the plasma, the erythrocytes, the leukocytes, the platelets, and so on. The question is, how are we able to study each? Okay? It is still this concept, this cell fractionation. Now, this one, it takes cells apart, even inside the cells. You can also separate major cell organelles and other subcellular structures from one another so that you can now easily study each independently. Is that okay?
Yeah.
Yeah, when I was teaching the Form 3's transport, one student was able to ask me that, but sir, how are we able really to separate these components of blood? And I was able to answer, but the answer was, imagine cell fractionation. I am sure this is your first time to hear the word.
Yes.
Or some books they call it centrifugation.
Yeah,
Centri.
that one.
You know that one,
Yes.
eh?
Yes.
Yeah. We use a centrifuge, okay, to separate these cellular components. And how does it do? It spins. Now you put your mixture of different cell organelles, different substances, different things, okay, in the test tubes, okay, and then you spin these test tubes holding mixtures of disrupted cells. The cells must be disrupted, not intact, okay. Or if they are organelles, or if there are any particles you want to separate, they must be disrupted, okay, not intact together at series of increasing speeds. Okay? Now, you must be knowing. Now, to do this, you must know the literature. That if I want to separate my organelles, the nucleus is going to sediment at this speed. So when you reach that speed, you remove. The Golgi apparatus is going to sediment at this speed. When you reach this speed, you remove. The mitochondria are going to sediment at this speed. When you reach that speed, you remove. Okay? Now,
Yes.
at lower speed, remember it is an increasing speed. So at lower speeds, the pellets that are large, or the pellets of large components, are going to be formed at the bottom of the test tubes. And so you remove those pellets. Okay? And those pellets will be having organelles that are large. Maybe if you know the sizes, the mitochondria has this size, so it will sediment at this speed. And usually the speeds are measured in S. S. S is it Sevadberg, something of that kind. Okay? We shall see. So if you know that the ribosome sediments at 70 Sevadbergs, 70S, then when you reach that 70S, you stop. The pellets that will be formed, they will be of ribosomes. Is that okay?
Yes.
Then, as you increase the speed, then the pellets with smaller components, they will be sedimented. And so, by that, you'll be able to separate these cell components, okay, into, you'll be able to isolate them so that you now study them as much as you want. So this is a centrifuge, okay? I don't know how many we have in Uganda, but this is a centrifuge. And so this is where, this is a compartment where you're going to put your test tubes, okay? Here, these are the test tubes. And so when you turn it on, now this is for, if you know that I want to separate, for example, ribosomes, and the ribosomes, they sediment at 80S, then you go here, you turn in 80. Then for it to sediment, when you reach 80, you stop. The ribosomes will sediment and then you come and isolate them out. Okay?
Yes.
So that is how it works. So this is the inside part. Okay? So it will spin. Okay? So this is before, it will be the same, but when you spin, then it will sediment and will form pellets. Okay? And so if this is blood, then that's how it is going to be. The liquid will remain up, then the sediments, the pellets will sediment at the bottom. Is that okay?
Yes.
Yeah. And so that is what